My name is Kaylee Arnold, and I'm a third-year PhD in the Odom School of Ecology. My research focuses on the interactions between the environment and the gut microbial communities of bugs that spread disease, specifically Chagas disease, which is found throughout Latin America. Long before I became a scientist, I began dancing and playing the cello when I was about 5 years old. I grew up in San Diego and fell in love with wildlife as my family would often take trips to the San Diego Zoo and the beach. After graduating college, I spent 3 months in Italy as an intern for a conservation group studying the ecology of resident bottlenose dolphins. I then moved back to San Diego and began working as a fish technician for an NGO, enhancing the coastal populations of white sea bass. During this time, I also volunteered with the San Diego Zoo's Institute for Conservation Research and discovered a love for wildlife disease research. By 2015, I began an MS program at Tulane University and studied the bacterial symbionts of bald cypress trees. Since moving to Athens, I am most proud of my burgeoning science communication skills and overall outreach. I am fully aware that I am a young Black woman in STEM, and there are not many scientists who look like me, particularly in the environmentally related sciences. I am the incoming president of an environmental STEM outreach group, EcoReach, and we often visit classrooms and the public library in Athens. If I can encourage even a few young kids of color to go into ecology or environmental sciences, that will make my journey and efforts in grad school so much fuller. I am a passionate young scientist who is working not only to increase the knowledge of a neglected tropical disease, but also working to increase representation in science and teach the public about what we as scientists do. Before I talk about my research, I first need to describe my study system, Chagas disease. Over 6 million people worldwide are infected, mainly in the Americas, and Chagas disease is a significant cause of morbidity and mortality in Latin America. Approximately 30% of infected people develop severe and often fatal heart disease. Chagas disease is caused by the parasite Trypanosoma cruzi and transmitted by blood-sucking insects, also known as kissing bugs. Specifically, I ask how landscape-level processes, such as deforestation, influence the gut microbial communities of kissing bugs, and how the patterns of these microbial communities may influence kissing bug survival and their ability to transmit Chagas disease. I focus on the gut microbial communities because they play multiple and critical roles. Landscapes are changing as the human population grows and expands, which has led to land use change as well as altered contact rates between humans, animals, and disease vectors. I study Chagas disease in Panama, where the main disease vector is the kissing bug species Rhamnius pallescens. Recent studies have shown that kissing bug populations increase in response to deforestation. However, we don't understand why this happens or if it causes other biological changes within kissing bug populations. Could it be that deforestation somehow leads to changes in the gut microbial communities of kissing bugs in ways that promote their survival, reproduction, or parasite infection prevalence? My doctoral research asks two main questions. 1. Does land use change in the form of deforestation alter the gut microbial communities of kissing bugs? 2. Does an altered gut microbial community have an effect on the kissing bug's ability to survive and reproduce? And vice versa, does a parasite have an effect on the patterns of the gut microbial communities found in these kissing bugs? Once we better understand the gut microbial community composition of wild kissing bugs, and the function of these communities within their guts, we can develop vector control approaches that aim to modify these gut microbial communities in ways that can reduce kissing bug survival, reproduction, or even Chagas disease transmission. I collected kissing bugs throughout central Panama using specialized traps to place in the crowns of royal palm trees, which we then retrieved a day later. Once returning to Athens, I dissected the mid and hind guts of the kissing bugs, extracted the DNA, and sent the samples out for DNA sequencing. So with this grant money, it'll be a lot of help with funds to travel to and from Panama while I finish up my fieldwork over the next few years, as well as professional development. So there's a lot of international conferences that tend to occur outside of the U.S. because they focus on global infectious diseases. And by attending these conferences, I'll be able to network with a wide range of scholars and public health researchers who are also working on tropical diseases, which, like Chagas, affects poor communities with limited access to water and sanitation.